Now I'm recording. Great. So welcome everybody. As I said, the final 2018 webinar for the California Master Beekeepers at UC Davis. It's great to see everybody. We have with us this evening Laura Brutscher. She's a postdoctorate with the El Nino Bee Lab, and I'm going to hand it over to Laura now. She can explain who she is and what she does much better than I can. So thank you very much for showing up this evening, Laura.
Yeah, thank you for having me, and thanks for, I guess everyone, thanks for watching tonight. Yeah, my name is Laura Brutscher, and I'm a postdoc in Alina's lab at UC Davis, and I've been here about 11 months. And before that, I was actually in Bozeman, Montana, getting my Ph.D. in microbiology. And there, my research focused on honeybee viruses, and that's kind of where my honeybee virus expertise comes from. And yeah, and before that, I grew up in Minnesota, grew up on a dairy farm. So you might hear a little Minnesota accent in there. Mm-hmm. All right. All right, everyone. I'm excited to tell you a little bit about honeybee viruses. So I'm sure, as most of us are aware, there has been high annual losses in honeybee colonies. So for example, the Bee Informed Partnership has been recording winter losses in honeybee colonies since 2006. And so our average winter losses have been around 26%. And then starting in about 2010, BIP has been recording total annual loss. So they've been looking at both winter losses and summer losses. And this number is a lot more startling in that since 2010, we've been experiencing an average of 37.8% losses in colonies. And so these losses are quite high and unsustainable. So the major question here is, what's killing the bees? Well, actually, there are multiple factors impacting bee health, including beekeeping management style, agrochemical exposure, nutrition. Nutrition's a big thing because many of these honeybee colonies are brought out to these monocultures, or just basically fields of one type of plant. And so the bees are only getting a single source of nutrition and pollen. And just like us, bees need a diversity in their diet. What I'll be focusing on tonight, however, is the role of pathogens and parasites in honeybee losses. In fact, high colony abundance and incidence have been associated with colony losses, as well as colony collapse disorder in colonies. And I'm sorry, my dog just walked in and is shaking. So honeybees can be infected by a wide variety of pathogens, including viruses, fungi, bacteria, trypanosomatids, and then they can also be parasitized by mites. But what you can see visually here is that the list of viruses is much bigger than that of any of these other pathogen groups. And so honeybee viruses encompass one of the largest groups of honeybee infecting pathogens. And it's important to note that, I have listed several pathogens on this page. I think as many viruses that we know of now, but honeybee viruses are being continuously discovered. And so the list is ongoing. It's growing as sequencing technologies improve, as detection technologies improve. It's also important to note that honeybees can be infected by more than one pathogen at one time. And so this makes studying honeybee pathogens all that more difficult. Because if we could just study one pathogen at one time in a colony and see how that affects colony health, that would just simplify science. But of course, it's not that way. So what are viruses? Well, to start off, viruses come in a variety of shapes and sizes. So for example, bacteriophages, which specifically infect bacteria, have this very structured spider-like look to them. Whereas viruses such as Ebola virus, which causes Ebola, they have more of this filamentous kind of stringy look to them. And then here's another another interesting example of virus structure where we have this tobacco mosaic virus, which infects tobacco and other plants. And it's hard to see here, but it has this very coiled-like structure. Honeybee viruses, in contrast, have what we call an icosahedral structure. So if you kind of look at this image of these little virus particles, they kind of look like little soccer balls. So despite the diversity in shape of these viruses, they all have a common theme to them. And that is that they're all these protein packaging or packages, so these shells here. And inside these protein shells are nucleic acid. So either RNA or DNA. So our genome is made out of DNA. Honeybee viruses can have a DNA genome. And other honeybee, or other, sorry, not honeybee viruses, viruses in general can have a DNA genome, but other viruses may have an RNA genome. And most honeybee viruses, such as deformed wing virus, are going to have an RNA genome. So there's kind of a question about, are viruses alive? Are they living organisms? And some people may argue that they're not, because they can't survive on their own. They need host cells to replicate. So let's say that this is a honeybee virus and this is a honeybee cell. During a virus infection, a virus is going to find a receptor, which is basically a protein poking out of that cell. And different viruses will specifically target different receptors. And what they do, one way that they can enter cells, that they'll bind to that receptor, and then they'll enter the cell. And then their DNA, their nucleic acid is released from their protein shell. And from there on, they replicate their nucleic acid, and then they produce more of their protein shells and some additional proteins that may go on the outside of the protein, or outside of the capsid. And then over time, the cell will then replicate, and often using the cell's the host cell's own proteins. And then at a certain point, they'll burst out of the cell so that they can go and infect new cells. So I'm going to give you a little bit of a molecular biology lesson. Stay there with me. But one of the ways that we can kind of understand virus structure and virus genomes is by looking at gene expression. So you may know that our DNA, our genomes, are made out of double-stranded DNA. So here we see one strand, and then we see another strand, and together they create this double helix. Our genome is double-stranded DNA. Honeybee genomes are double-stranded DNA. And so DNA in itself doesn't really do anything, but rather it serves as a blueprint for proteins in our body. And the proteins are what really are important for all of our body's processes. Our metabolism, cellular regeneration, hair growth, like proteins are all important for that. And so in order for this blueprint, our DNA blueprint, to become proteins, it first needs to transcribe into RNA, which is the single-stranded molecule. And then from there on, the RNA is then translated into protein. In contrast to us as humans or honeybees or dogs, RNA viruses get to skip this transcription step because they already have an RNA genome and they can directly translate that RNA into proteins. So it gives them a little bit of a head start when infecting a cell. So back to the basics. When we think of viruses, we think of them as being very, very tiny. So here is kind of a colorful scale of all the shapes and sizes that we might find in life. And here we're focusing on things we can see by our own naked eye, and then things we can see using a light microscope. So that's a microscope that you might find in a microbiology lab. Well, you can use microscopes to look at cells and bacteria. Most viruses are just too small. You can't see them underneath a microscope. So you can use this specialized equipment and expensive equipment known as an electron microscope to look at viruses. Well, rather, you'd probably have to pay someone to look at viruses. So just to give you another example of how small viruses are, here is a single shaft of a human hair shaft. And we can see that compared to the shaft of hair, viruses are extremely tiny. And you can imagine that how many viruses could coat a single piece of hair? A lot. So what sort of diseases do honeybee viruses cause? Well, some honeybee viruses can cause symptoms in larvae. So for example, sac brood virus causes this sac-like, it causes larvae to look like a basically dried up old sac. There's also black queen cell virus, and that will specifically target queen larvae and cause them to blacken and darken. So while these viruses cause these symptoms in developing larvae, adult bees can also have them too with no obvious symptoms. When we talk about adult symptoms or adult diseases, we probably all are aware of deformed wing virus, in which deformed wing virus causes deformed wings in adult bees, adult and developing bees. And then another example of an adult viral disease is chronic bee paralysis virus. So this causes the bees to lose all their hair, and it kind of gives them this shiny, greasy appearance. And it also affects their mobility and prevents them from being able to fly, and essentially slowly paralyzes or slowly immobilizes them. And again, I want to say that not all virus-infected bees exhibit obvious symptoms. If you go into an apparently healthy colony and screen for honeybee viruses, you will find some. You will definitely find some. And I would almost argue that more often than not, you can find a virus without it causing any obvious symptoms. So honeybee viruses are everywhere. So I also wanted to touch upon routes of virus transmission. So there are similar routes of transmission as to us and humans. For example, bees can transmit viruses mouth to mouth or via trophallaxis. So, bees are often transferring nectar to each other from their honey stomachs and communicating to each other mouth to mouth. Another route is the fecal-oral route. And then another route is foodborne. So if you were to go into a colony and screen their pollen or nectar, you could probably find honeybee pathogens in those food sources. Another route of virus transmission is sexual transmission. So just like us, honeybees have sexually transmitted diseases or infections, excuse me. And basically honeybee drones can transmit pathogens to queens. And we don't know for sure, but it's very likely that drones can transmit deformed wing virus to queens. So another route is vector-borne. So for example, varroa mites are a major vector of honeybee viruses, predominantly deformed wing virus. And then lastly, this is something that not many people think about, and it's still a very active area of research, of an actively growing area of research, is that honeybees can transmit pathogens to other species. So for example, a study in Great Britain has found that honeybees and bumblebees that share the same floral sources can transmit deformed wing virus to each other. So let's see. So there's all these honeybee pathogens. There's so many ways to transmit viruses. And you know, how do we even know that we're there? How do we detect honeybee viruses? Well, as I mentioned earlier, we can use transmission electron microscopy. And here are just a couple example images of what you might see. So here's an image of black queen cell virus and deformed wing virus. And then here's an example of deformed wing virus. And as you can see, in this one picture, you can't really tell the particles apart from each other. TEM isn't very useful in terms of showing you anything other than viral particle size and shape. And it's not very helpful in terms of differentiating viruses from each other. And to compound the issue even further is that if you look at poliovirus, which have the same shape and size, they also have the same shape and size to deformed wing virus and black queen cell virus. So TEM is not a very reliable method of looking for and identifying honeybee viruses. So what honeybee scientists do is that they use molecular biology. So again, we're looking at this picture of DNA, RNA, and proteins. And again, most honeybee viruses have an RNA genome. And the way that we can detect viruses the most accurately is by screening for their RNA. And we have a few different methods in which we can do that. We have polymerase chain reaction. We can use microarrays, and then we can also use high throughput sequencing. So polymerase chain reaction is really useful if you know the sequence of the virus that you're looking for. And yeah, it's basically useful if you know what you're looking for. And so you can actually develop what's called a primer to target a specific region of your viral genome. So let's say deformed wing virus, and we want to detect like a specific sequence that's specific to deformed wing virus. You can't find it anywhere else in any other virus or any other sequence in any organism. We can then use PCR to target that sequence, and then we can amplify that sequence so much, we can amplify that nucleic acid sequence so much that we can actually visually detect it on a gel. And PCR is also really useful in terms of giving you amount of the viral sequence that's in there. So you can quantify virus using PCR. Another method that was more common in past years, but it's not very common anymore, is that you can use a microarray, which is basically this little slide with little pieces of DNA on it that are specific to different nucleic acid sequences. So for example, let's call this a pathogen microarray. You know, we can say this circle is DWV, this is Israeli acute paralysis virus, this is ABPV, and they're all specific to that specific virus. And so using this method, we can take a sample, you know, a honeybee sample, we can take all the RNA, convert that to DNA from that honeybee sample, spread it across a microarray, and see if any viral sequences attach to these oligonucleotides. And if we see attachment, then we can say, like, yes, that pathogen is present. And it can also give us a sense of the abundance of that pathogen. So microarrays are kind of they're on their way out. And more commonly, we use high throughput sequencing. And this is really great because you don't have to know what you're looking for. All you have to do is have your nucleic acid sample, and it will sequence everything in that sample. And then based on the sequences that it outputs on your computer, you can then match them up to the known genomes of different pathogens to see if what sequences those, or what pathogens those sequences belong to. So this may be going a little too far, but RNA must be converted to DNA before using any of these methods. And so we can actually use a special enzyme called reverse transcriptase that's actually found in other viruses, retroviruses, that will actively convert RNA into DNA. And that's better to use because DNA is much more stable than RNA. All right, next I thought I would talk about studies correlating pathogen incidence and abundance with colony health. And here is a large list, not fully comprehensive, but a good start of studies that have found a connection with pathogens and colony health. And so this is just for you to look at later if you're interested in. But for now, I'm just going to focus on one study, on one more recent study that I'm a little more familiar with because I know the authors, William Glennie, who was a master's student with me at Montana State University, and then in Michelle Flanagan's lab, which is the lab I worked at Montana State University. So in this study, they tracked migratory colonies that had just left, that were in California, for the almond pollination season, that were then transported to Minnesota later in the year. And they tracked mite loads of these colonies, and then they also collected samples from each of these colonies over time and screened these samples for several different pathogens. And this is what you'd call a longitudinal study, where they followed the same colonies over these several months and tracked their pathogen abundance and presence. And what we can see on this chart is, yes, it's very complicated, but we can see the pathogen diversity of each colony. And what you can see is that each colony is infected with definitely more than 3 pathogens at one time. And I'm just gonna move something. There we go, okay. And what we can see is that there's a bit of seasonality within these viruses, within these pathogens. We can see that starting in June, all colonies started to have deformed wing virus. And we can also see that stopping in about June, colonies up until then were infected with Lotmaria passum, which is a small eukaryotic pathogen. It's related to Cryphidia. So this like for the field of longitudinal honeybee virus studies, this is one of the better ones, I would say. They also looked at pathogen. They also looked at DWV abundance and its relation to mite infestation. And what they found was that the higher the amount of mites per colony, the higher the likelihood the colony would have a higher DWV abundance. They also, this is also a common theme in a lot of longitudinal studies, is they looked at to see if there were any correlations between different pathogens and their abundance. And once again, they found that Varroa destructor mites were positively associated with DWV. So this table is a little complicated, but all you need to do is pay attention to the numbers in bold. And if you see a positive number, that means that there is a positive association, meaning that both go up at the same time. And if you see a negative number, that means that that when one is high, the other is low. So for example, again, we see a negative association between Lotmaria passum and deformed wing virus, which is similar to that graph we saw earlier, where as soon as DWV showed up, we hardly detected any Lotmaria passum. And it's hard to say why that is, if deformed wing virus like actively excluded Lotmaria passum infection, or if just seasonally Lotmaria passum falls off during that time of year, so then that was DWV's turn to opportunistically take over. And it's really hard to say because, again, this is a correlative study, so it's we can't make any assumptions about that. So, so I briefly covered one longitudinal study, and then I wanted to zoom in into honeybees and tell you a little bit how honeybees respond to virus infection. What do they do to fight off viral infections? And so honeybees have both individual antiviral defense mechanisms and they have social antiviral defense mechanisms. So one example of that is that bees create propolis and they'll line their colonies with propolis. And propolis in itself has antibacterial properties. Antimicrobial properties. In terms of it being specifically antiviral, we don't really know yet, but I know that Marla Spivak's lab at the University of Minnesota is looking into that. Another social immune mechanism is called hive fever. So hives will actually, if they're experiencing an infection, they'll actually heat up the hive, just as we would have a fever while we're sick, in order to help rid that pathogen. From now on, I'm just going to focus on individual antiviral defense. And I'm going to talk about some of my grad work. All right, so I'm going to throw a bunch of acronyms at you, and luckily this is recorded, so you can listen back if you get confused, or pause, or just fast forward, whatever you want to do. But just like us, like mammals, honeybees actually have pathogen recognition receptors. So that's what that acronym is, pathogen recognition receptors. And what these do is that they'll actively detect and respond to pathogen-associated molecular patterns. So an example of a pathogen-associated molecular pattern is double-stranded RNA. Honeybee viruses although most of them are single-stranded RNA, when they're infecting a cell, in order to replicate their RNA, they actually will copy on another strand onto their RNA, and in that process creating double-stranded RNA. And insects have, and so this is a pathogen-associated molecular pattern that insects can detect and initiate an immune response to. And there are several types of immune responses that insects can have. So an example is cellular responses. Just like us, honeybees have specialized cells, which are called hemocytes, that can target pathogens. So in this case, this is an example of a cell phagocytizing a pathogen. And what it will actually do is engulf the pathogen and then digest it down with digestive enzymes, efficiently killing off the pathogen. Honeybees also have cells that can surround a pathogen if it's too big to swallow, and that the cells can then release oxidative chemicals that will break down the pathogen. There are also humoral responses, so that means we're focusing on a single cell and it has individual cellular pathways that it's initiating. And it's a single cell can release different proteins outside of the cell to help fight off pathogens. One of the primary antiviral defense mechanisms that we know of in insects is RNA interference. Most of RNA work has been examined in fruit flies, which are distantly related to honeybees. But basically, RNA interference is a post-transcriptional gene silencing mechanism, and it's sequence specific. So what I mean by that is if we have, let's call this a honeybee virus. If we have a honeybee virus infecting a cell and then replicating, making that double-stranded RNA, the cell has a way to detect that double-stranded RNA and respond to it, called RNAi. And so what happens is that it will actually, proteins will bind to the double-stranded RNA and break it down until eventually it's the single stranded molecule that can help this enzyme bind to and detect similar sequences and to cleave it. So essentially, once it has double-stranded RNA from, let's call this deformed wing virus, it can then use this single-stranded beacon to bind to other pieces of deformed wing virus genome and break it down. Yes, let's see what else I have to say about it. I think that's it. So we know that this is the primary antiviral defense mechanism in fruit flies, but there's still more to be learned about its role in honeybees. We know that treating honeybees with double-stranded RNA that's specific to a specific honeybee virus can actually reduce virus load, which that's really neat. But we also know that double-stranded RNA, when it's non-sequence specific, can also initiate antiviral defense in honeybees. And so my graduate work really focused on understanding what this non-specific pathway was. What's happening in addition to RNAi in honeybees? So while I was in Michelle Flanagan's lab at Montana State University, I investigated antiviral defense mechanisms in honeybees. And I basically infected bees with virus and also co-treated bees with double-stranded RNA that was either specific to the virus, so that would be an RNAi mechanism, and then I also injected them with double-stranded RNA that was not specific. So if it had any effect, it would have nothing to do with RNAi. And what I found was that, yes, treating bees with both types of RNA, both types of double-stranded RNA, whether it was sequence specific to the virus or sequence non-specific, reduced the virus load as compared to our bees given virus alone. So this y-axis is virus load, and this is the treatment. You don't need to worry about these guys. So my next step was, I was really interested in what are the proteins, what's going on in this non-sequence specific pathway. And so in order to do this, I utilized high throughput sequencing. So specifically, I utilized RNA-seq to look at what genes honeybees were turning on in response to virus infection. And so what I mean by turning on, what genes were being actively transcribed into RNA. And so I used high throughput sequencing, And so I collected RNA from each of our bees and then performed sequencing on these bees. And the basic answer, the overall conclusion is that it's very complex what plays a role in honeybee antiviral defense. Interestingly, I identified this not very well characterized gene, and it seemed to be the most highly expressed in all of our virus-infected genes. And we still don't know very much about it, but studies are ongoing in the Flanagan lab in terms of describing and understanding this gene. So I'm glad to be a part of, to have kind of launched future studies in this gene. Interestingly, when looking at bees that were treated with non-specific double-stranded RNA, I also saw an increase in these two RNA helicase genes. And this is interesting to me, at least, because the RNA helicases tend to bind to RNA, and these could be honeybee-specific RNA helicases that are specific to double-stranded RNA in honeybees, which we've never discovered these, or we've never identified these as playing a role in honeybee antiviral defense before. So these are also good targets for future studies in honeybee antiviral defense. I also wanted to talk a little bit about the role, a little bit more about the role of mites in honeybee antiviral defense. And I wanted to highlight this really interesting study that looked at DWV strain diversity in honeybees. And so, you know, we call DWV, but really there's a few different strains of DWV, meaning that their genetic sequences are just a little different from each other. And what this study found is that what the study was able to do was look at DWV diversity in colonies that were not affected by mites. And so they focused on colonies based in Hawaii before the mites could reach these colonies, or these islands, basically. And this was a really cool, kind of cool but sad time-lapse study in terms of what happens to DWV diversity as mites take over honeybee colonies. And so they tracked this island as well as Maui and Big Island. And what they found was that in 2009, where there was no mites, or very few mites to be found, that the strain diversity was very high. And also the DWV prevalence, or the number of colonies infected with DWV, was low. So diversity was high, prevalence was low. But throughout time, as the mites started to become introduced onto these colonies, they saw a decrease in DWV strain diversity and then an increase in DWV prevalence. And so it's kind of interesting that as mites started to enter these colonies, there seemed to be the single strain of DWV that seemed to overtake all the other strains and then infect the bees at high levels. Another study, so kind of working off of this, so basically we can summarize from this that there might be some synergistic effects between mites and DWV. Another study that kind of springboarded off of this, so to say, further examine the role of mites in DWV diversity. And they looked at deformed wing virus as well as Varroa destructor virus 1, or it's also known, Varroa destructor virus 1, which is very, very closely related to DWV and can also be vectored by mites. And what they found is that colonies, again with no mites, had low levels of both viruses and a greater diversity of both viruses. They then looked at colonies with a low levels of mites, and they found slightly higher levels of the viruses, but still, and then slightly reduced diversity. And then when they looked at colonies that were high in mites, we saw way, way, way greater levels of both viruses, and they actually detected this recombinant strain of the two. So what I mean by recombinant is that something about the mite transmission of these viruses made these viruses develop or evolve, well, not evolve, but develop into this kind of intermediate strain that had sequences from both viruses, and it was present in the bees at high loads. And so, and so they were interested in like, what were the mechanics behind this? Why was this happening? And one thing they did was that they physically injected pupae in the same way that a might might inject DWV into pupae. And they found that had the same effect, that injecting the bees with this diverse pool of DWV and VDV resulted in this high level of this single strain of virus. So it suggests that there's something about the mite transmission, something about the way that the mites physically infect the bees that results in this more virulent strain of DWV. And then lastly, I wanted to talk about nutrition in antiviral defense. And this is an older study, but I think, actually I skipped one. I have two more things to talk about. I wanted to talk about the role of agrochemical exposure in virus levels, specifically, and then nutrition. And what we found with agrochemical exposure, or at least these two studies have found, that bees given imidacloprid and clothianidin, when given increasing doses of these two chemicals, had increasing levels of DWV, which indicates that that these chemicals, while not directly fatal to bees, it can affect their immune response to viruses and make them more susceptible to virus infection. So these are what we would call sublethal effects. This additional study also looked at bees given, not treated with anything, bees treated with black queen cell virus alone, bees treated with thiacloprid, and then bees treated with a combination of both. And what they found is that there's something about this chemical that when given with black queen cell virus increases black queen cell virus loads. So this further exemplifies that, even though agrochemical exposure, and these were given at what they'd called field level doses. It's really hard to say what is field level dose for a bee. But this shows that lower doses of these chemicals can be harmful to bees, even if it doesn't directly kill them. I also wanted to say that not all studies show this connection. So I picked out these two studies, and you might think it's all doom and gloom, but I've also been involved in other studies where they've done similar experiments where they've infected bees with virus and given them increased doses of different agrochemical exposures. And in the one study I was involved with, in some cases they found no correlation. In some cases they even found decreased virus load with higher doses of agrochemicals. So it's definitely not as clean cut as these two graphs that I'm showing you, and that more research needs to be done in terms of the role of agrochemicals in honeybee health. All right, so next I want to talk about the role of nutrition. And as I said earlier, it's really important for bees to, have a diversity in pollen and get enough pollen, which is a really good source of protein for bees. So this study didn't directly look at pollen diversity, but they did look at the role of pollen versus protein supplements versus bees only receiving sugar water. And what they found is that bees that were given sugar water, so all bees were given sugar water, but bees that were just given sugar water and no protein had higher levels of DWV as compared to bees given protein supplements and then bees given pollen. So this exemplifies the role of nutrition, specifically protein, in honeybee antiviral defense and health. And that would be all for me. If you have any lingering questions after tonight, I would be happy to answer your questions within reason. And don't overload my email box, but if you really have a pressing question, I'd be happy to see if I can help clarify it for you. But with that, thank you so much for hanging in there with me.
Thank you, Laura. It was a very, very informative talk. I'm unmuting some folks, so if anybody is interested in asking Laura any questions, please just chime in. And if you can just mute some of your background noise, that would be much appreciated as well. Thank you so much. Any questions from anyone?
I have a question. Hi, Laura.
Hello.
Alina.
Hi, Alina.
Hi, everyone else. Thanks, Laura. That was great. And also, I just wanted to say it was a great review of the advanced anatomy and a little bit of physiology class. And I should also tell all of the people who are here that if you're going to take the journey level exam, you can expect some of this material to be on it. So I'm glad you're here. But I do have a quick question about viruses. Well, two actually, that I think would probably be of interest to people. First, the question that I get often is that, well, the first one is, how long can viruses be viable on equipment? Because clearly they can survive, although they're not really alive. But they can be viable in pollen and nectar, you said, for a while. Do we know anything about the length of their viability on equipment?
Not that I know of. I don't think I've seen any studies on that topic. Some people I know that work with viruses will say, like, just how fragile viruses are, and like just trying, it's funny when you want things to work, when you want your virus inoculum to do an experiment, it just doesn't work, like it's degraded. But at the same time, the fact that honeybees and bumblebees can transmit viruses with each other when they're sharing floral resources, that somewhat argues to the point that some viruses are stable for a time. But in terms of specific studies looking at equipment, I don't think there are any.
That means you should do some.
Yeah. On top of everything else.
On top of everything else. Not next year, but the year after, because you're not going anywhere, Laura. Another question that I get often is, what about vaccinating bees for viruses? So you mentioned that if you feed them double-stranded RNA, right, it activates RNAi and there are fewer viruses. So I know that some companies are working on it. But what about the other thing where you have the multiple DWV strains? And I believe there's one that's more virulent or more prevalent than the other. I can't remember now. They're what, A, B, C or something like that?
Yeah, I think B is the most prevalent.
Yeah, but A is not as virulent, right? It's not as bad for them. So if you were able to somehow introduce higher levels of DWV strand A, although nobody would agree on to do any of those studies probably. But is there anything, you think there's any utility in that?
So now that you mention that, it kind of it reminds me of the study, and I might butcher it, but I think they were longitudinally monitoring colonies, and they found these colonies that were predominantly infected with, I think, type A, strain A of DWV, and they found that, I think the sample size was small, and we need to go back and read the paper, but I think they did find a small correlation, or did find that these colonies that were first infected with strain A were more resistant and less likely to be infected with a more virulent strain.
So do you think that could potentially be used somehow in the field?
I think that would be hard, because I don't know. I honestly, I suppose, I suppose anything's possible. I suppose that's the next experiment I could do.
There you go. You're catching on, Laura.
Yeah.
What about RNAi?
Oh, right. So Biologics, which is a company that was bought out by Monsanto, they're not selling it anymore, but they were selling these cocktails of siRNA targeting specific viruses. And the research in terms of the results in terms of its effectiveness towards reducing virus load was actually not very clear. There's one study on it. They found that colonies receiving this cocktail produced more honey, but the study never really looked at virus load. So that makes me believe that it probably did not really did not impact virus load very well.
Yeah, interesting. Thank you.
Yeah, thanks, Alina.
No problem.
Any other questions?
Yes, I do. This is Terry.
Hi, Terry.
Hi. I'm wondering, how many bee viruses have we identified up to now?
It's about in the 20s range. I don't have the precise number because, like I said earlier, like new bee viruses are being constantly discovered by sequencing, but I would say it's in the 20s right now.
Okay.
Let me just interject. I think there is one paper that just came out from Grosinger lab. I don't know, Laura, if you saw it.
Probably not.
It was really recent, like fresh off the presses. I think they identified at least another 26 from what I remember headlines. So it almost doubled the original amount
I'll be
that
darned.
we thought.
Yeah.
But we don't know what they do, right?
Yeah, I mean, so many of these viruses, we don't know what specific symptoms they exhibit, if any.
Of the viruses that you are familiar with, what percentage would you say are fatal to the colony versus maybe how many sort of kind of auto-correct themselves, or they kind of go away and not harmful to the colony?
When it comes to viruses, that's really difficult to answer because at any one time you'll just find numerous viruses with no obvious symptoms in the colonies. I mean, if I would have to guess,
Harry's asking questions.
I would say that deformed wing virus, in combination with mite infestation, is probably the most harmful to bees.
Okay. And then do all the viruses, or do most of the viruses have detrimental effects on the honeybees? Or did you just kind of answer that with?
I can rephrase it. I can, well, so there's every colony, whether it's healthy or not, are going to have pathogens in it, and and some colonies will even have high levels of pathogens, and it won't be like apparent to us. But yeah, I mean, like deformed wing virus at high levels, that's going to have obvious symptoms. Israeli acute paralysis virus is going to have is going to have some obvious symptoms. Chronic bee paralysis virus, but again, a healthy colony may have all of these viruses in it, and you wouldn't know it by looking at it.
Okay. And I guess it's because I was thinking, like with the bee gut, you know, they have beneficial and then the pathogenic microbes. And so I was just wondering if maybe some of the viruses weren't, you know, as virulent, or I don't know what the right word is, but maybe there's some, they're not as effective as other.
Yeah. Yeah, some studies have shown. So, well, this is going to be a roundabout way of answering this. But yeah, the honeybee gut microbiome is important for antiviral defense. I think more studies need to be done on it, but but we know that treating bees with antibiotics can make them more susceptible to other bacterial infections, and I think some viral infections as well. So yeah, and I guess what I mean by virulence in this instance is that the virus replicates so that there's a lot of virus to high levels in the bee. So that's my answer for you.
Thanks.
So Laura, what do you think about this new discovery that the mites feed specifically, well, they're targeting feeding on fat bodies? How would that change our way of thinking about how to treat these things?
That's a good question, Alina. Yeah, it's hard to say because, yeah, traditionally, or before we knew that mites fed on fat bodies, we could just say, like, oh, it feeds on hemolymph, so the viruses enter through the hemolymph.
Mm-hmm.
In terms of like the mites feeding on the fat bodies, I'm not sure like what that implies, if the viruses are first infecting the fat bodies and then, you know, being being transmitted into the hemolymph.
By accident, yeah.
Yeah, it's hard to say.
Interesting. Thanks,
Again,
Laura.
another study I need to do. Just kidding.
Yeah.
Any other questions? Oh. All right. Well, you've given us a lot to consider. And
That's one way to put it.
well, and I wanna say that the way that you've presented it with the graphics and going slide by slide, it was very helpful to me as a layperson. I'm not trained in science. I was able to follow along, so I really appreciated the knowledge that I've gleaned out of your presentation this evening.
Well, thank you.
And I'm sure that everybody else will concur. I'm looking forward to being able to share this on our Camp Listserv and upload it onto Facebook. So, and I thank you so much for your time, Laura, this evening.
No problem. Thank you.
Yes, and keep on researching, please. Yes, we need you. Thanks for showing up.
Thanks, Laura. Thank you, everyone.
Thank you. Bye.
Bye.
Some of you, I hope to see some of you at CSBA next week, maybe.
Yes. Yeah, for sure. Take care.
Bye.
Good night, everyone.
Bye.
Good night.
Good night.